you could potentially not do as well as you'd like. Okay, so yes, Monday, Wednesday, people who have me for lab, your test is on Wednesday. Tuesday, Thursday, people, your test is on Thursday of next week. Okay, so I am going to, oh, go ahead. It'll be the same as last time, about 30 to 40 multiple choice, and then it'll weigh about 30%, and then the discussion questions will be the other 70%. That's how I did it last time, 30-70. You can do the practice test for extra credit, and you can do the an extra discussion question for extra credit. So those of you that aren't getting 80s or above on the discussion questions, consider doing the extra question, because that helps a lot of people for not getting full credit on one question. They make up for some of those points with that extra credit question. And just so you know, if you don't tell me which question is the extra credit, I automatically assume it's the last question. So, and it's only worth half the point, so it's worth 10 points where the other ones are worth 20. So if you had an intended extra credit, make sure you indicate that to me somehow. All right. Okay, so when will practice test for 4 be up? Is it not up now? I will make sure it's up today. I don't see it on here easily for me just to do. So I'll put that up today. Let me make a note to myself. If I don't make a note to myself, I will forget.
Dr. Bird.
Huh?
I resubmitted the DQ3. Did you see my email?
Who is this?
Kriti?
Oh, you resubmitted it to me. I don't know if I got it, so I will look for it. Okay. I'm not sure if I went back in and did it, so I'll look for it. Thank you for reminding me. And maybe send me an email.
Yeah, I did. I emailed you like last week.
Okay. I will look for it. I'm sorry if I missed it.
Oh, you're fine.
I've been getting a whole bunch of those emails where you guys are trying to get into the Google shared drive and trying to get in with like your other Gmail accounts. So I get like 50 of those in a row. So sometimes I lose an email that might be amongst those that I'm thinking are all of those emails. So I apologize. I did post the discussion questions. You should see them. It may take a second to load. Let me see. I see them there. So try again, Gretchen. See if you can see them. Yeah, I see them in both sections. Yeah, okay.
They're on there.
You see them? Okay. Mm-hmm. Sometimes I think maybe there's a little bit of a lag between me and what you see. I don't know. All right, so we are going to finish chapter 13 and head into 14 hopefully today. A lot of times I roll 15 into 14, so if we don't really get to 15, we'll just have to go without it, because a lot of what's in 15 I can kind of teach as I'm teaching 14, so just so you know that there's a little bit of a combination there. I haven't posted my voiceover for 14 yet, but that's coming. Okay, so we are going to, hold on, I can't see my PowerPoints. That's what always happens. We are going to finish chapter 13 right now. Let's see. Can you see my screen? Yeah? I think you can.
Yes.
Excellent. Okay, so we talked about the basic idea of DNA replication last time. The general gist of it is the strands separate. Each one is used as a template. We build a new strand. So our goal now is to really find out how this works with all the enzymes involved and what it really looks like in the cell. And I'm going to go through kind of these still pictures pretty quickly, and then we're going to watch a video which I think helps see it in real time, can really be useful for understanding all of this. Okay, so we already talked about origins of replication, and we talked a little bit about these enzymes. So let's just start here, review the enzymes. So the first thing we have to do is pull apart the strands. That's done by an enzyme called helicase. So think of it as the enzyme that unwinds the helix. So that's where it got its name. Then we have to make sure those single strands don't go push back together. So we have to have single-stranded binding proteins that basically hold them apart and stabilize them. Because we're untwisting here, we get over-twisting over here. So we have to have something fix that overcoiling. That's called a topoisomerase. That happens kind of farther away from the origin of replication. And then if you remember, our polymerase is persnickety and requires a nucleotide to be present on the new strand in order to start the new strand. So a primer has to be put down on the DNA first before the polymerase can do its job. And the primer we use is built by an enzyme called primase. It's shown there in pink. All right, and then of course the enzyme that does the actual building of the new DNA template is called DNA polymerase. And like I said, it's very persnickety, and these are the things you need to remember. It can only read the parent strand. This is reading, not building. It can only read in a 3' to 5' direction. So if you go back to this picture here, remember our strands run in an anti-parallel fashion. One strand runs where the 5' is pointing one way and the 3 is the other way. The other strand runs in the opposite direction. Are you going to have to recognize which is which? No. But do you need to know that they run in anti-parallel fashions? Yes. And you need to know what direction polymerase works? Yes. All right. So it can only.
In our book,
Yes, go for it.
sorry, I was just gonna say in our book it always shows that the lighter color is the new one. Is that like a thing? Is that something that happens everywhere?
That's very common. I can't say that it happens in every picture that you look at, but yes, typically it's the lighter one. So it can only read 3' to 5', and if you know that, then you can just infer from that that the one that's being built is built in a 5' to 3' fashion. So yes, Brock, that's exactly right. And then the other persnickety thing is it can't just start de novo. That's what that means. It can't just start from scratch to build this new DNA strands. It has to have a nucleotide already present. So that's why we have to have the primer to start with. Okay, so I have my cursor on that screen. Why is it not going forward? Okay. So, okay, if we're going to see what this looks like, here's our replication, origin of replication. Here's our replication bubble. Now think about those two dark blue strands, those original strands, they run in opposite directions. One of them is 3' to 5', the other one is 5' to 3'. All right? So from that origin, we're going to build the new strands moving in both directions on both strands from that origin. However, only in one direction on each side is that original strand actually going in the 3' to 5' direction. So you can see here, this little red thing represents the primer. And, oops, can you see that? Yeah. And then this blue one is the new strand being built. All right. Which means when we're going on this strand, we're reading this in a 3' to 5' direction, and we're building this one in a 5' to 3' direction. Same things going on here on this side of the origin on this strand. And then what you see is on the other side of the origin on both strands, it doesn't go in a continuous fashion. It still has to go 3' to 5'. So you can see the arrows are still pointing in the same direction. But if we're starting here, we go here and then we bump into this primer. So then we have to go back and start here, and then we're going to bump into this primer. So we have to go back and we start here, and we're going to bump into this primer. And the strands are opening up. Oh, it's so frustrating that I can't show you. But the strands are continuing to open, going in this direction, and then again in this direction. All right? So because of the persnickety nature of DNA polymerase, we can't build the entire new DNA strand for both strands in a similar way. We have this one part that's continuous, and we just keep building, building, building. That's that long blue arrow. And then we have this other part that gets made in these little teeny fragments. They're called Okazaki fragments. Okay? So these right here are called, they're named after the guy that discovered them, Okazaki fragments. And then this one's just going to keep going till the end of the chromosome on that one. Okay? These red things are the RNA primers. So of course at some point we're going to have to go in and remove these RNA primers and replace them with DNA nucleotides because they're RNA and we don't really want a DNA RNA hybrid at the end. We want a complete DNA piece at the end. Okay, so we're going to, when we are done, we're going to rely on another DNA polymerase that's going to come in and remove the primer and then replace it with DNA. And then we're going to end up with this little piece, these two pieces of DNA that aren't connected. Because it's a polymerase, it can't connect them in this way. So then another enzyme called DNA ligase, and the word ligate means to join. DNA ligase will bring those two pieces together and make that discontinuous strand continuous. So another polymerase, a different version of the polymerase, comes and takes away the primer, replaces it with DNA nucleotides, and then has to connect the two pieces together. That's done by the ligase. All right, so there's a lot of enzymes that you got to know what they do. Helicase, primase, the main polymerase, the other polymerase, and that you can, if you look in the book, I think the main polymerase is DNA polymerase 3, and then the ligase one's DNA polymerase 1. I'm sorry, not the ligase one, the one that replaces the RNA primers, and then then ligase you have to know as well. Okay, so let's just think about this, given what we just talked about. Here's a piece of DNA. You can see the direction 3' to 5'. Now, assuming primase works like polymerase, and what that means is it runs in the same way in terms of reading the strand 3' to 5'. If the primer is to start at that T that's underlined, what would the primer sequence be? So go ahead and think about that for a second. I'm going to turn my fan on because I'm hot. Okay. So if it only runs 3' to 5', which direction are we going to go? Are we going to go in this direction, or are we going to go in that direction to build the primer? To the right or to the left?
Go to the right, right?
Got to go to the right. It reads 3' to 5'. So kind of like left to right.
5 to 3.
Okay, good. So what would the primer sequence be? That's a good start. Let's just say we go all of these nucleotides. Let's do all of them just so we know that we're doing it properly. So what would pair with the T? Not a U. What pairs with T? An A. Good. Then what pairs with the G? A C. Oops, I thought someone put a C and it said Cyrus. Sorry. I was looking at your name, Cyrus. So the G would pair with the C. What would the C pair with? A G. Good. How about this A? U. There you go. U again. And then what would this T pair with again? A. Okay, good. I think you guys got it. So you can see what I mean when I say we move from 3' to 5', and then you make sure with that primer, you're thinking in your head that it has to be RNA, and then you have to make the correct pairings between. T always pairs with A, G pairs with C, C pairs with G, and then A pairs with U. So don't just replace the T's with U. You actually need to pair them.
Does the primer run across the whole DNA strand or only one point?
It's about 10, somewhere between 7 to 10 nucleotides long, something like that.
Okay, so if we were to answer a question, we would just assume it was from that point to the end?
Yeah, I just wanted to go to the end because we had a chance to do all 4 nucleotides. I didn't want to start with the first because there was no A.
Okay, sorry, just making sure.
That's okay. All right, so here we go. Here's that 3' to 5' piece of DNA. Now the primer is reading along, adding it. Notice how the new one that's being built is 5' to 3', right? Then polymerase will come and bind. Starting off of this last nucleotide from the primer, it's going to be able to add new nucleotides, which are blue, right? And then when it reaches the next primer in line, it's going to dissociate, and it's going to kind of move backwards and start from the next one back. And that's how we get those fragments. All right. And then, of course, we have to replace the RNAs with DNA and then join the fragments with ligase. All right. So here's my ligase joining those fragments. I promise the video is much a better way of looking at it than still pictures like this. All right. Let's answer some questions first. So thinking about this image, predict which Okazaki fragment was made first. Okay, I got a C. Does everybody agree with Brock? You can tell this was designed to have my note cards, right? The colors. Does everybody agree with Brock, or do you think A was the right one? C, Juan says C. C. Okay, you're right. C was the first one, because remember, the bubble starts here. So the first one's going to be there, and let's go, oh, boom, and then it has to jump back. Oh, boom, and then it has to jump back. Oh, boom, and then these strands are continuing to be separated. Excellent. All right. So, and there might be more than one answer for this one. So looking at where things are labeled, there's an A, B, C, and a D. Tell me where the 3' ends are correctly labeled. So pay attention. If the D is at the arrow, that's saying it's that end versus on the red side of it. Which one is the 3' end? Okay, I have someone saying A, someone else saying B. So it can't be A and B, right? So let's think about that. Let's look at A and B, right? If this is reading in this direction, so that has to be the 3' end over here. This has to be 5', so it can't be A, right? So B has to be the right answer, because look how it's going in this direction. The arrow's going in that direction. So 3' has to be over here. All right, does everybody see that for the A and B, the difference? All right, now let's look at these two. Are either of these representing the 3' end? What direction are the new strands being made? 5' to 3'. So this is the 3' end. What about this one? So you're right. The C is the 3' end. How about the D? Is D 3' or is that 5'? Nope, this D is also 3'. Notice it's at the same arrow as that one. All right, so if this is the 3' end, that has to be the 5' end, which makes this one the 3' end. You got that one, and Cyrus? Okay, does that make sense? It is kind of sneaky. So you really have to kind of, when you're answering questions like this, kind of just breathe and take questions, slow down, just start marking out where things are. It's easy if you do it from the origin and figure out the parent strands, and then you should be able to figure out the other things. And keep in mind, the new strands are made 5' to 3'. So the arrow, the direction it's going, is always going to be that 3' end. Okay. I'm going to skip this for right now because we're going to watch it in a video, which is just easier anyway. Here's what the video is going to show you that those still pictures didn't show you. The polymerase doesn't actually jump off and then run back to the next spot and jump back on and start making the next fragment. In reality, the polymerases are kind of stabilized in one position. They're kind of clamped in a stable position. And what's actually moving around is the DNA. The DNA actually loops around and turns itself backwards on the strand that's not going in the right direction to make it go in the right direction for the polymerase. So we're going to see this in the video, and it's useful to see when you actually see it going on. Okay, so a couple of vocabulary things before we go on. The one that has the Okazaki fragments is called the lagging strand, only because it obviously can't be made quite as fast. because it has to do all this movement around. It has to, the DNA has to move around. We have to replace more primers, that kind of thing. And then the strand that's going in a continuous fashion is called the leading strand. Now, when you read in the book, there's a lot of talk of the sense strand and the anti-sense strand. I will just tell you that I'm not going to make you know all of those words right now. For now, I think if we can get the concept down, after you understand this, later you can go back, probably in another biology class, and apply those terms to what's going on. I just feel like it's too many vocabulary words for right now, so you can just dismiss sense and anti-sense, and for right now, we're just going to leave it to leading and lagging. Okay, so let's go to our video. Let me see. I had it up, but then I ended up closing it, so I apologize. Give me a second. All right. All right, now pay close attention to this. It really is something that when you see it, it just starts to click and make sense. All right, here it goes.
Every cell in your body is produced by cell division. Before each cell divides, it must copy its genetic material in a process called DNA replication. Understanding of DNA replication comes largely from studies of E. coli, bacteria that are found by the billions in your large intestine. Let's take a look at how DNA replication occurs in an E. coli cell. As we zoom in, we see the DNA. At the origin of replication, the two strands of DNA separate, serving as templates for making new strands. The result is a replication bubble. The bubble grows in both directions, forming two replication forks.
Okay, I'm going to stop that. So what we normally see in the pictures, because I can't really show you, is just half of the bubble. So if I were to cover up the other half, right, you only see the Y, so half of the bubble. That's what they're calling the fork. So all those pictures we saw kind of showed half of the bubble. So just keep in mind that there's always the other half.
Let's zoom in on one of them.
Now we're going to the fork.
Many proteins work together at the replication fork. Only some are shown. Here, the DNA is unwound, and DNA polymerases, shown in orange, build new strands of DNA. Original parental DNA strands are shown in dark blue. Newly formed DNA strands are shown in light blue. Because strands in a DNA double helix run in opposite directions, the new strands must be made in different ways.
All right, let's watch that
One
again.
New strand,
I want you
the
to
bubble
see
grows in both directions,
the DNA
forming two
loop back
replication
around
forks.
on that bottom strand.
Let's
Watch
zoom
it
in on
zoom
one of
around,
them.
back around.
Many proteins work together at the...
I'm going to mute her. Okay, so you're going to watch this. This one's now building. Here's the primer that got laid. Here's the polymerase. It's going to build, 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 and then it's going to run into this primer. All right? When it does, the polymerase lets go. It can't connect them. It doesn't want to yet. Let's go and watch the DNA. It pulls back through. The polymerase really stays still. The primase is right here, pink. It's going to say, oh, make a new primer. And you can see it building the new primer down here. And then this is going to attach to the polymerase, and the polymerase is going to do that same process again. Everybody see how that worked? So the DNA moves, not the enzymes. You're right. The enzymes are kind of scaffolded in place, right? Okay, so I'm going to let her talk again. So now she's going to look at the continuous strand, the
First,
top strand.
let's focus on the leading strand. DNA polymerase builds a new strand of DNA by adding DNA nucleotides one at a time.
Okay, let's just quickly think about what is when they're building this new polymer of nucleotides, what's being connected from one nucleotide to the next? So think about what your nucleotide looks like. What's connected along the backbone? Silence from the group. Sugars and phosphates, exactly. Okay, and what kind of bond connects them? What bond forms between the sugar and the phosphate? Okay, add more phosphodiester bond, exactly. And what kind of reaction is this? Let you think. Okay, dehydration synthesis. What's another word for dehydration synthesis? Anabolic, good. And is this exergonic or endergonic? Endergonic, which means it requires energy, and it uses energy in the form of, to do this, ATP, right? Okay, so I just want to make sure you're not just seeing what's happening, that you're actually putting this all together. Like, this is why we learned all of those other things, so that now we're actually applying it to the real way it's being used in the cell.
Each new nucleotide must pair up with its complementary nucleotide on the parental strand. Adding new nucleotides works the same way on both the leading and lagging strands.
That little starburst was the ATP every time.
Each piece of the lagging
The other
strand.
thing I want to say is, you notice that the enzyme actually changed shape, right? That was a purposeful shape change that had to happen for every single new nucleotide that was put in. Purposeful shape change based on probably being activated by ATP to change that shape.
Strand begins with a short segment of RNA, shown in red. A clamp surrounds the RNA and attaches to DNA polymerase, which builds the rest of the new piece as DNA.
The clamp is just there for stability, to hold everything in place. You can almost think of it like a coenzyme. It's just kind of making sure everything is staying where it should be.
When the piece is finished, it is released from DNA polymerase. How are pieces of the lagging strand joined together? A different DNA polymerase removes RNA and replaces it with DNA. However, it cannot finish connecting the pieces. An enzyme called DNA ligase joins the pieces together.
Okay, so DNA ligase is a really common laboratory enzyme. So if you're ever wanting to make a hybrid DNA where you're going to combine a human gene into a bacterial cell and you need to put it in there, that ligase is really important. So you'll probably see that again somewhere else in your life.
Growth of the leading and lagging strands continues on both sides of the replication bubble until there are two identical DNA molecules. Although bacteria are very different from humans, the process of DNA replication in bacteria is similar to what happens in your own cells.
The only thing that's really different is the enzymes are a little bit different sized. They are sometimes a little bit different shaped, but they functionally do the same thing. All right, so I want to know if you can tell me, looking at this picture, what would be the difference between this nucleotide right here and that nucleotide right there? How does the cell distinguish between an RNA nucleotide and a DNA nucleotide? Okay, the sugar, right? So the red one has what kind of sugar? Ribose, and then the blue one has what kind? Deoxyribose, right? Okay, now that video is not one that you can see because you have to have access to the textbook. So if you bought that, you can see it that way. This one is free, so everybody can see this one. And so I want to make sure that you have one that you can actually look for and watch again. So if you go to HHMI, which stands for Howard Hughes Medical Institute Biointeractive, and then type in the search bar DNA replication. They have lots of great videos here. So you can type in anything you want and look at their videos. We're going to watch this one. She's far more boring. I much prefer the lady from Pearson. But what I like about this one is going to show you basically the same thing. Just keep in mind everything's flipped-flopped. So on that last one, we had the lagging strand on the bottom. We're going to have it on the top this time. But she shows it to you kind of in real time, and you get a sense of how absolutely crazily fast this can happen. Okay, so here we go. Is she going to talk? Oh, I have her muted. Sorry.
Copying occurs in the local.
Let me start over. I didn't know I had her muted.
During DNA replication, both strands of the double helix act as templates for the formation of new DNA molecules. Copying occurs in the localized region called the replication fork, which is a Y-shaped structure where new DNA strands are synthesized by a multi-enzyme complex. Here, the DNA to be copied enters the complex from the left. One new strand is leaving at the top of frame, and the other new strand is leaving at the bottom. The first step in DNA replication is the separation of the two strands by an enzyme called helicase. This spins the incoming DNA to unravel it at 10,000 RPM in the case of bacterial systems. The separated strands are called 3' and 5', distinguished by the direction in which their component nucleotides join up. The 3' DNA strand, also known as the leading strand, is diverted to a DNA polymerase and is used as a continuous template for the synthesis of the first-order DNA helix. The other half of the DNA double helix, known as the lagging strand, has the opposite orientation and consequently requires a more complicated copying mechanism. As it emerges from the helicase, the lagging strand is organized into sections called Okazaki fragments. These are then presented to a second DNA polymerase enzyme in the preferred 5' to 3' orientation. These sections are then effectively synthesized backwards. When the copying is complete, the finished section is released and the next loop is drawn back for replication. Intricate as this mechanism appears, numerous components have been deliberately left out to avoid complete confusion. The exposed strands of single DNA are covered by protective binding proteins, and in some systems, multiple Okazaki fragments may be present.
Okay, so you can kind of see how that was in real time, and it's pretty intense, right? I mean, you really can see what's going on. So hopefully that helped you visualize this picture and the idea of those Okazaki fragments. And I don't really like when things are named after people, but that's one of my favorite ones to say. I just think Okazaki is such a fun word to say. Okay, so let's answer this question. The difference in the leading versus lagging strand synthesis is a consequence of A, the larger size of the lagging strand, so does it have more base pairs? The physical structure of the DNA, because it's anti-parallel. The activity of the DNA polymerase, the direction it can move. The activity of the topoisomerase, the ability to break the two chromosomes, or are two of those correct? Okay, Marissa says 2 are correct. Which 2, Marissa? Or someone. Marissa doesn't want to answer it. B and C, that's right. Due to the fact that they have anti-parallel nature and the polymerase can only run in one direction, it can't possibly be made in the same way, right? So it's like if you live on a one-way street, you can't access if you follow the law, right? You can't access your house from both sides equally. You have to go in the one way always, right? And go around the block the other way. Okay, Now, polymerases are pretty darn accurate. You would think that polymerases might make a lot of mistakes given how fast this all happens, but they really are very, very accurate. It's very low, but it's not zero, so it does sometimes make mistakes. There's a whole family of different kinds of DNA polymerases, and there's another one that is his job is to do proofreading. They all have numbers like polymerase 1, 2, 3, 4, 5. One of them is used for proofreading, and basically goes behind the building polymerase and proofreads. Did the bases match up appropriately? So sometimes it might find a mismatch. Maybe the A got paired with a T somehow, somewhere, right? Then that polymerase will recruit repair enzymes and correct it. Sometimes you might get things like this. Sometimes when there's two T's right next to each other, they like each other a little bit more than they like to hydrogen bond with their A counterparts. And they actually form a covalent bond between the two T's. It's called a thymine dimer, okay? So the proofreading mechanism goes along, and recognizes, because it actually kind of bubbles out of the DNA a little bit, makes it look funny. And so call in a nuclease that will cut the DNA, and then another polymerase will come in and replace it, and then a ligase will come and join it all back together. All right? So lots of things are in the cell to kind of reduce the number of possible errors that could be made. And remember, are all changes, are all mutations bad? Like if we left one and didn't fix it, is that always a bad thing? No. So let's talk about some mutations that probably wouldn't be very bad. Name me one kind of mutation that probably wouldn't be very bad. Anybody? Okay, the CCR5 one from the one that we talked about that gave us immunity to HIV, is that the one you're thinking of? Okay, that's good. So I'm more thinking just in general, but yes, you're right, because that would be one that for natural selection purposes is useful, right? So yes, not mutations are bad because some of them actually benefit us in the event of natural selection. So Elaine says silent mutations. So silent mutations might be something that change the DNA, but don't actually end up changing the protein. All right, so we'll talk a little bit more about how that works when we get to the next chapter. But yeah, you can have a change in the DNA that doesn't actually affect the protein. And that would be if it was in the gene. But you could also have mutations outside of the gene. There's a lot of DNA that's not part of genes. So for example, you could have a mutation in the centromere region or a mutation in the telomere region that aren't genic. They don't have any DNA, any genes in their DNA anyway. And so if you have a mutation there, it's not necessarily going to cause a disease or something like that. Excellent. Okay, now the ends of our chromosomes, have we talked about telomeres before? That's the really highly heterochromatic ends of our chromosomes. Have we talked about them? I can't remember. Okay, yes. So basically, at the ends of our chromosomes, we run into a little bit of a problem on those lagging strands because we have now some primer that doesn't end up getting filled in. All right, so that last primer, 5, 6, 7 base pairs, doesn't get filled in. So with each round of DNA replication, our chromosomes get 5 to 7 nucleotides shorter every single time we do DNA replication. This is why we have that protective telomere at the end of our chromosome that doesn't have any genes in it, because if we can protect that from getting shorter and shorter over time, if we don't have any genes right here, if we get shorter and shorter and shorter, it's not going to matter. It's not going to affect genes. It's not going to affect proteins. It's not going to affect the central dogma, and we're going to be just fine. All right. However, if they keep shortening and they shorten into the region of the chromosome that has genes right here, then we are kind of in trouble because now we're going to potentially be deleting nucleotides that are parts of the gene. All right. So telomere shortening is something that's considered a component of aging, because as your telomeres get shorter, you're getting into the genic part of your chromosomes. And does anyone know how we make sure we don't have shortened telomeres when we pass our chromosomes on to the next generation? I'm not sure whether we talked about that or not before. Yeah, new ones get made. So there's an enzyme in our germ cells, and it's only active in our germ cells, that's called telomerase. So it's the enzyme that builds telomeres, and they rebuild the length of that telomere before we pass that chromosome on to the next generation. So you can imagine that that could be a possible anti-aging kind of activity if we could somehow activate telomerase in cells that are aging, like in your wrinkle cells, right? If we could maybe get the chromosomes to be a little longer and not shortening into the genes anymore, could we use that as an anti-aging? It hasn't really gone anywhere, but it still has potential. Okay, so so don't forget our conversations about euchromatin and heterochromatin, right? So those are important things to keep in mind while all this is happening. When DNA replication happens, we have to kind of get in that heterochromatic mode and get rid of all those histone proteins because they're in our way. But as soon as we do the replication, we start winding back up around those histones and making it heterochromatic again. Remind me, when does a cell do DNA replication? That's right, during interphase, during S phase. And what do we call the resulting structure that comes out of DNA replication? What does it look like to us? I'll go to the next picture because maybe that helps. What do we call it after it's duplicated? Well, it does eventually get to be heterochromatic during mitosis. That's true, but not right away, not till mitosis. So what we get, okay, one of them is a chromatid, you're right, and then the other one is the other chromatid. The result of DNA replication is that duplicated chromosome, that X structure that we've seen before, that has one old strand and new strand as one chromatid, and the other old strand and new strand as the other chromatid, and remember they're connected together at the centromere via those cohesin proteins that we've talked about. Okay, so which of the following statements regarding DNA packing is false? A nucleosome, which I didn't really tell you that word, consists of DNA wound around a protein core of 8 histones. I'll tell you that's true. That's the definition of a nucleosome. DNA packing promotes gene expression. Histones account for half the mass of eukaryotic chromosomes. Highly compacted chromatin is generally not expressed at all. Or prokaryotes have proteins analogous to histones. Which one's false? Nobody knows? I told you A was true, so you know it's not A. What ones are you thinking about? B, that's right. B is false. Remember, the more packed it is, that's the less gene expression we have. That's more heterochromatic. Good. So B is the correct answer. All right. Keep in mind about the change that chromatin undergoes during the cell cycle. So euchromatin is the relaxed chromatin that we see during G1, S, and G2. Euchromatin promotes gene expression. Heterochromatin is what we see definitely during mitosis. And then there are other regions like the centromere and the telomere that pretty much stay heterochromatic all the time. And so that's what I was just saying. The parts that stay heterochromatic all the time, those are telomeres and centromeres. So we're going to talk a little bit about this when we get to the next chapter, so I'll just introduce it to you right now. So those histone proteins that the DNA wraps around have tails that part of the protein kind of has a little tail, and modifications on those tails will influence the architecture of that chromosome, whether it's really tightly wound up like this, more heterochromatic, and the gene here, this is the gene I'm interested in, is inaccessible. I can't get my proteins in there to do their work. And then I can change the modifications on those histone tails, and that opens up the structure and allows me access to that gene. All right, so histone methylation promotes condensation, and I think we talked about that before because those methyl groups are hydrophobic, so they attract each other and they make it all kind of clump up. And then another thing called acetyl group causes it, that has an oxygen on it, that causes it to open back out. All right, so you should know the difference between a methyl group and an acetyl group. All right, so if we want to convert euchromatin to heterochromatin, so we want to go from being loose to being tight, what will we do? Will we involve a helicase? Will we involve acetylation of the histone tails, methylation of the histone tails, or will you use a repressor protein? And we haven't even talked about that yet. What's your answer? Yes, methylation is what's going to promote euchromatin to heterochromatin. All right, so we can, by just coming in and changing what modifications here will actually influence the change in the architecture. Okay, and notice when the DNA is then accessible, right, you see right here, you can have polymerase bind and things happening. Now, if we look at these histone tails, boy, they have been studied to a lot. This is a field called epigenetics, which basically means a change in your genetics in terms of your change in your traits that doesn't have to do with a change in the actual DNA sequence. Because if you could just change the histone tail modification and make a gene accessible or not accessible, you can very much influence whether the gene is expressed or not expressed. So people study this quite a lot. So there's a lot of residues along the histone tail that can be modified. They're mostly lysines. So there's a lysine at position 4 that can be methylated. There's a lysine at position 9 that can be methylated. There's a lysine at position 27. And it's really kind of a symphony of events that has to happen to really get exactly what you're looking for. So sometimes you might have to have a combination of a variety of different things present in order to get the outcome that you're looking for. Okay, so that is the end of chapter 13. Do we have any questions on 13 before we hit to 14? So we're going to kind of keep going. We're going to come back to that histones at the end of 14. All right, so chapter 14, we finally made it to my favorite chapter of the whole semester. This is the chapter on the central dogma. We're finally going to work out all the nitty-gritty details of how the central dogma really works. If you get the question about the central dogma wrong on the test, I will cry many tears because you should know this by now. So no getting this wrong, okay? We know that the central dogma is the process of taking the information within the DNA, a gene, copying that information into a disposable copy that doesn't have to stick around for very long in an RNA fashion, taking that RNA out of the nucleus to a ribosome. The code on the RNA is read by the ribosome to build the primary structure of the protein, and then that has to go through its own folding process and transportation process to get where it needs to go. All right, so let's remind ourselves, what's a gene? Go ahead and type it in the chat. I need like Jeopardy music. Nobody knows what a gene is? Oh, come on. Okay, it's a hereditary segment of the DNA. It's a unit of heredity. All right, now give me a unit of inheritance. Now give me the definition I gave you. A section of the DNA that does what? What does it have in it? A distinct sequence of nucleotides that has the information, right, to code for a particular protein or a particular RNA, right? That's the important part. And I'm not picking on anybody, but these other answers, the hereditary segment, unit of heredity, those are like everyday people definitions for a gene. You, as biologists, need to know more specifically what that gene does. It's coding for function, right? Either a functional protein, and we're going to learn in this section, or a functional RNA. Okay, how do we name genes? Do you know? That's not something we've talked about. Anyone know how we name genes? They're named in a variety of different ways, really. Sometimes they're named for a disease they cause. So, for example, the breast cancer gene, it's actually called breast cancer gene. It was found to be associated and makes people, if they have a certain mutation, it makes you more susceptible to getting breast cancer. So sometimes it's named after the disorder or the disease, I'm doing that for chat, that it might cause, right? Other times it's named for the protein that it produces. So for example, this is an easy example, the hemoglobin protein comes from a gene that we call the hemoglobin gene, right? Or the p53 protein comes from a gene that we call the p53 gene. So you have to be careful. There are some real specific kind of grammar rules when it comes to genes. So for example, when you're talking about the gene of the DNA, typically you do that in lowercase. If you're talking about the if they have the same name, like p53, then you would do it in uppercase, but then sometimes you don't have that option like with p53. So you have to kind of just learn the nuances of that when you're reading. All right, what does the term gene expression mean? What would be another way to say gene expression? You guys are super quiet today. Is Ivan here? Even Ivan's being quiet. He may not be here. Is that why? Because Ivan's not here? He was here, I thought, earlier. He must have had to leave. All right, so nobody knows? Okay. So another way of saying gene expression is the central dogma. This is how DNA codes for proteins. So when we talk about gene expression, we mean getting to the end product where we have a functional protein out there doing something in the cell. Now, different types of cells in our body express different types of genes and make a different subset of proteins. All right, so we have to kind of understand how and when that happens. So different genes make different proteins, and different cells have an accumulation of different proteins through what's called differential gene expression. So what this means is all the cells have the same DNA. My blood cells have the same DNA as my skin cells. However, I don't express the same genes to make the same proteins in those different cell subtypes. For example, in my blood cells, I want to make hemoglobin. So I'm going to express the hemoglobin gene. I don't need hemoglobin in my skin cells, so I'm not going to turn that gene on, which means I'm not going to do transcription of it in my skin cells. So the cell actually has very specific control over which genes are actually being expressed or made into protein for each and every cell. All right. So here, for example, here is a liver cell. Here is a neurotransmitter from me. Okay, so they have exactly the same DNA. They have exactly the same DNA. All right. In a liver cell, I need to express a protein called alcohol dehydrogenase. All right. So that means the alcohol dehydrogenase gene needs to be expressed, made into RNA, and made into protein so that I can have the functionality of that protein in those cells. In a neurotransmitter, in a neuron, I need to make neurotransmitter proteins, which means in my neuron, I need to have the neurotransmitter gene expressed on to make RNA to make these proteins. However, the other in the opposite cell types are needed to be turned off. I don't want alcohol dehydrogenase being expressed in my neurons. That would be a big problem. I don't need my neurotransmitter to be expressed in my liver cell. So we differentially express the genes that we need, and we don't express things that aren't necessary in the moment. Keep in mind that the genetic code is universal. So one of the things that we're going to be talking about when we take this idea of going from gene to protein is we can take the gene from one organism and put it into a different organism, and it's not going to know the difference. So for example, this is a tobacco plant that is expressing the firefly. So you've all seen fireflies with the little butt that glows. The gene that allows for the butt to glow. So the whole plant glows. This is a pig expressing that same protein, right? So you can see how his nose looks yellow and his hooves look yellow. So this is like a firefly pig, which is pretty cool. And you can see his little neighbor back here doesn't have the yellow nose, right? They didn't make that one look like that. And this is all because of DNA universality and then the central dogma. So someone asks, is this gene editing? Yes, this would be gene editing. This would be more like cloning a gene from one organism and then expressing it in a different organism. So they didn't necessarily change a gene. They just plopped it out of one organism, plopped it into another organism, and had it express it. All right. So obviously this idea of the central dogma that we have literally talked about every single day since the first day of class is super important. All right. It happens a little differently in prokaryotes versus eukaryotes, but essentially the idea is the same. The only really big difference is that in eukaryotes we have a nucleus that's in the way. We have way more genes, way more proteins being made, so we have a little bit more kind of control maybe over it. All right, so before we get into that discussion of how it works, we have to have a little bit of background so we know what we're talking about. First of all, we're going to be talking a lot about RNA, different kinds of RNA, not just the one that is part of the central dogma. So I want you to be sure you know the difference between an RNA nucleotide and a DNA nucleotide. So we know the U and the T differ, the ribose and the deoxyribose differ, the length differs. DNAs are long, thousands of genes long. The RNAs are the length of a gene. It's the only length that they are. The DNA pretty much stays in the nucleus all the time. The RNA actually leaves the nucleus every single time. The RNA is single-stranded. The DNA is double-stranded. All right. So make sure you are aware of those differences. They are very great test question material, right? Okay, now I will say that sometimes single-stranded RNA folds back on itself, and we're going to see an example of that later. So it kind of looks like it's double helix, or it looks like it's double-stranded, but it's really not, still just one strand. Okay, so there are different kinds of RNAs. The kind that we've been referencing this whole semester when we talk about DNA to RNA to protein is actually called the messenger RNA. So that particular protein part of the central dogma, that is not just any RNA, that's the messenger RNA. It takes the message and delivers it to the ribosome to be made into a protein. Messenger RNAs are always translated into proteins. These are the only ones that are translated into proteins. We're also going to talk about what's called transfer RNA. So this is one of those that you can see it folded back on itself. So this is still just one strand, but because it found complementary pairing with itself, it folded back on itself. tRNAs are coded by a gene. So there's a tRNA gene in the DNA. However, in this case, that gene codes specifically for this functional tRNA. This tRNA never gets made into a protein. It functions as an RNA. All right? Its job is to help the ribosome translate the message from this mRNA into amino acids. So we'll learn how that works a little bit later. You've probably heard of ribosomal RNAs. These are the RNAs that are part of the big ribosome complex, which is both protein and ribosomes. Again, the ribosomal RNAs, that's not really the shape of it, that's just the whole ribosome there. But the ribosomal RNAs are coded for by a gene, transcribed to make an RNA, and then they stop there and they function as an RNA. So can you tell me something you might know about where do ribosomal RNAs get transcribed? This is putting together some of the things we've learned earlier this semester.
I'm
Okay.
assuming. Oh.
Go ahead, Mark. Okay, be more specific. So yes, in the nucleus of a eukaryotic cell. Where in the nucleus? That's right, in the nucleolus. Remember, we learned about that part where there's active transcription. Those are the RNAs that are going to be rRNAs part of this. And then the RNAs that are messenger RNAs that become ribosomal proteins are also transcribed there. Excellent. There are other types of RNAs. In fact, there's a lot of other kinds of RNAs, and they are extremely useful in the cell. So, for example, there's microRNAs that help regulate gene expression. There are small nuclear RNAs. We're going to talk about them in a little bit. There are lots of other kinds of RNAs that function as RNAs, meaning they don't get made into protein. The only one that ever gets made into protein is this messenger RNA. Okay, any questions on that before we move forward? So genes code for proteins via mRNA, but genes also can code for RNAs that function as an RNA. Okay, so the first part of all of this is transcription, where we're going to take the code in the DNA, the gene, and make a copy into RNA. So how does, let's answer the second question first. How does transcription differ from replication? So we just learned about replication. How does transcription differ from replication? Where's Ivan when we need him? I'll have to email him and tell him I missed him. Okay, transcription is used for not just synthesizing proteins, but for synthesizing RNAs that are going to become proteins or RNAs that are going to be functional. Okay, they make RNAs. Replication produces an identical set. You're right in the sense that it has T's and not U's. Okay, replication is the permanent synthesis of another copy, while transcription is just a brief copy that's going to come and go. Okay, so I thought maybe you were going to go on what we just talked about and what this picture shows. In replication, we copy both strands from end to end, right? The whole thing. In transcription, we only copy one strand, and we only do it for the length of the gene, and then we're done. So it's a much shorter process, a lot easier, doesn't involve all that looping around and Okazaki fragments and all that stuff, because all we're doing is we're just copying one strand, and we're just going to copy the strand that goes in the right direction. So that just totally makes sense. Okay, so how does this really work? The two strands have to separate. Now, we don't use a helicase. In this case, the RNA polymerase pretty much does all of the work. So the RNA polymerase binds, and it just starts to kind of put some torque on the DNA and unwinds it. All right. The two strands will separate right at the beginning of the gene. All right, and only one strand is used as a template. All right. So I kind of think I already said it, but can you guess the name of the enzyme that builds RNA? Anybody? RNA polymerase, exactly. RNA polymerase, that's exactly right. The amount of DNA that's copied, that's just the length of the gene, is building a new RNA endergonic or exergonic. Good, excellent. All right. And then we have to keep in mind the complementary base pairing rules between DNA and RNA, right? So A pairs with U, T pairs with A, G pairs with C, C pairs with G. So keep that in mind. RNA polymerase is the enzyme, and it's very much like DNA polymerase. It's persnickety in the sense that it can only read in one direction, same direction as DNA polymerase. However, it's not persnickety in the sense that it doesn't need a primer. It can jumpstart this reaction from the get-go, similar to like how primase worked. Remember, primase just went in there and started building an RNA. RNA polymerase can do that as well. Shape-wise, it's the same. It kind of clamps on, and then it just kind of reads one nucleotide at a time, and brings in the appropriate nucleotide. So the strands are going to be separated by the polymerase. The polymerase is going to read along and build the new RNA strand using complementary base pairing rules. It's going to know where the gene starts, and it's going to know where the gene ends, and we're going to talk about that in just 1 second. And it's going to release the RNA, and the DNA is going to go back to a hybrid as soon as it's done. All right, so as soon as the RNA is being made, it pulls off of the DNA goes back into a helix, and the RNA kind of falls off of it like a tail. All right, when it gets to the end of the gene, the polymerase knows it. The polymerase isn't in this picture. It's going to then release the RNA altogether, and the DNAs will go back together via complementary base pairing. Okay, so the most probably important part of transcription is making that call whether we should do this or not. Do we do transcription or do we wait? Do we need this right now? Do we need to spend this energy? Do we need this protein function or not? All right, so we have to first look at what the gene looks like so we can understand how that control works. All right, so every gene has basically 3 parts to it. All right, the first part of every gene is called a promoter. It's not actually part of the gene. It is the sequence right before the gene starts, or the important information starts. So it's kind of like if you want to get on, 35, and you're on, I don't know, the service road, you can't just jump on. You have to go via an on-ramp. So think of the promoter like the on-ramp. This is how we get to the highway. This is how we get to the important information of the gene. Then that's going to be juxtaposed right next to the coding region. This is actually the sequence that's going to dictate the protein that's going to be made or the RNA that's going to be made. So now we're on the highway. The coding sequence is the highway that we're going to be traveling on. And then when the coding sequence is done, when the gene is actually done, we have to get off the highway. So there's an exit ramp, okay? That's called the termination sequence. All of these are DNA. All of these are a sequence of nucleotides. It's just these sequence of nucleotides don't code for protein. This doesn't code for protein, although they're informative nucleotides that tell the polymerase where to start and where to stop. And then in the middle, these ones actually code for amino acids. And the steps of transcription follow along these parts of the gene very nicely. All right. Now, I want to talk a little bit about promoters. This is that on-ramp to the gene. This information, a little bit of what I'm telling you right now, comes from chapter 15, but I'm pulling it in right here because I just feel like it fits right here, okay? So the promoter is DNA. I can't say that enough. That's something that confuses people a lot. The promoter is a sequence of nucleotides in the DNA. It's before the gene starts. It's the on-ramp. There are proteins that can bind DNA, and the Specific proteins that bind promoter DNA are often called transcription factors. So transcription factors are proteins that bind to promoter DNA sequences. And the transcription factors have to be present in order for us to switch whether the gene will be transcribed or not. Okay, so we kind of are going to be able to turn the transcription on and off via these transcription factors. The transcription factors are controlled by other proteins that bind other places on the DNA. All right, so there are, this is the picture I want, sorry. So my promoter's right here. So here's my gene in red and white, okay. Here's my promoter right upstream from it. This is all DNA sequence here. All right, and then here's part of the promoter. The promoter's pretty long, all right. And then far away from the promoter, not necessarily right next to it, it can be thousands of base pairs away, are other DNA sequences called enhancers. Okay, so let me say this. Enhancers are DNA sequences to which other proteins can bind. All right, so if we were to look at a piece of DNA, this represents DNA right here. Here's my gene in kind of dark blue. My promoters here in green. And then I have these enhancers that are far away. And notice here I have different enhancers. This is how I can control gene expression in various different cell types. Proteins will bind to these enhancer sequences and flip the switch for the transcription factors to be on or off. All right, so I can have the transcription factors here at this promoter and maybe a little bit of transcriptions happening. I can have the transcription factors at the promoter here in this cell type with this extra protein bound to this enhancer and get more gene expression. So now I can have it up higher levels. Or I can have the transcription factors here, but I can have an inhibitor bound here that's going to really slow this down and we're hardly ever going to express this gene or turn it off completely. So we are controlling on and off and level by the combination of proteins that are present between the promoter and the enhancers. Okay? Okay, so regulatory proteins bind DNA at places called control sequences. So I've talked about a couple so far. I'm going to introduce some more. Transcription factors are promoters that bind, transcription factors are proteins that bind to the promoter. Activator proteins are ones that turn transcription up, making it happen more. They bind to those enhancer sequences. Repressor proteins, remember we had that in that question a couple questions ago. Repressor proteins are proteins that bind to DNA sequences called silencers. So that was like that red protein in that last picture I just showed you. And it's, again, it's almost like a symphony. You have to have everything present at the right time in order to make this movement into transcription happen. And it involves kind of a really complex event that involves DNA bending. All right, I like the next picture better. All right, so here's my coding sequence here. This is the start of my gene right here, the blue. This is my promoter. So you can see polymerase is here waiting at the promoter to get started. And then all of these ones with numbers and letters are part of the transcription factors that bind the promoter. So the polymerase and the transcription factors bind to the promoter and to each other. They kind of stack up all on top of each other. And then far away from the promoter can be an enhancer to which an activator protein can bind. The DNA actually will bend when it needs to to bring the activator protein into contact with the transcription factor. When that happens, then the transcription factors basically kind of pull off the polymerase and tell the polymerase to go. Go ahead, do transcription. We got the final signal that it's time to do it. And what you don't see in this picture, this repressor protein, if it were to bind right here, it might stop this bending of the DNA so that the transcription factors aren't getting the full signal, and the polymerase will then slow down its activity or stop it altogether. So there's a whole bunch of stuff that has to happen before we ever even get to the start of the gene. The gene, we haven't even gotten to the start of the gene yet. We're just on that on-ramp. But in order for us to get on that on-ramp and actually get onto the highway, we have to have all of these components in place to make that happen. Marissa, what's your question?
So is all of these sections of the DNA to activate this process, is all of that called the operon? Because I know it's one of our vocabulary terms that I know we haven't gotten to yet.
Good question. Okay, so we're not going to get to operons today. That'll probably be next Tuesday. Operons are only in bacteria. What I'm teaching you right now is how it works in eukaryotes, and it's very different in bacteria and eukaryotes, and I'll tell you why. There's a lot of extra DNA in eukaryotes that's not gene, that serves as regulatory DNA like enhancers and promoters. In prokaryotes, there's very little extra DNA. In fact, the genes are oftentimes, there may be 3 or 5 genes under the control of one promoter. In eukaryotes, every gene has its own promoter and enhancer and silencer. Every gene has its own. So this is what I'm showing you now is not operons at all, and we're going to get to it. There are some similar components, but we're not talking about operons yet. And Ivan, we just saw that you came in and we missed you because nobody's been talking all class.
Sorry about that. I totally forgot today.
That's okay. I'm just glad you're here. Now you can talk for the next 5 minutes. We have about 5 more minutes. Okay. So this is a really cool process that the DNA actually bends, and we have to have all of these things come together. And keep in mind, how do we know when to bring the activators in? Well, that's part of that whole cascade of events. So maybe you get a growth factor that binds to a receptor on the cell surface. And then that receptor protein gets activated and has kinase activity and phosphorylates something, that phosphorylates something else, that phosphorylates something else, that phosphorylates this activator, which enters the nucleus, which binds the DNA, which causes the DNA to bend, and then associates with the transcription factor. All of that has to happen just to get this one gene to be transcribed. So it's under some very tight control. This is not willy-nilly. We don't spend time making RNAs we don't need. So this only happens in the event that we need it. However, when it happens very precisely, very accurately, and extremely quickly. Because once we do this and we activate it, we can keep making new RNAs over and over. We can make, from one activation signal, we can make hundreds of RNAs. Right? Once we get the signal, we can make lots. Does that make sense?
Yes.
All right. Yay, Ivan's back. Okay, so the stages of transcription fall in line with the parts of the gene, right? So the first part of transcription is called initiation. That's when all of that stuff has to be established, and the RNA is sitting in the promoter waiting for that go-ahead signal. Once it gets the go-ahead signal, then it goes into the next step of transcription, which is called elongation, and that's just the building of the RNA through the coding sequence. And then the last part of transcription is termination, when the polymerase reaches a sequence within the DNA that's called the terminator sequence. And it says, okay, polymerase, you're done. RNA, you're going to get released. DNA, you just go back to being your own thing. So you can see how they match up with the 3 parts of the gene. Initiation matches with the promoter, elongation with the coding sequence, termination with the terminator sequence. And we can have, like I said, lots of polymerase doing this. Once we get the activation signal, polymerases can keep binding to that promoter and starting and starting and starting. So we can be making 1, 2, 3, 4, 5, 6 from that one activation event at a time. All right, the termination part of this is just basically a sequence at the end of the gene. All right. Once we, well, let me talk about this first, and I'll talk about this picture. Once we get to the terminator in a eukaryote, the RNA is not quite done yet, and we're going to have to kind of cook it a little bit more before we really let it out to be made into protein. In a prokaryote, we take the RNA, even as it still may be bound to the DNA, and start translating it right away. Because there's no separation of nucleus to cytoplasm, the ribosomes are right there, so they can just start to do translation right away. So what I'm trying to say is translation in a bacteria can start before we even ever get to the termination sequence. That's not the case in eukaryotes. In eukaryotes, you have to get fully to the termination sequence, which is typically a whole bunch of A's, A. It keeps going a little bit past those A's and then says, okay, I'm going to let go. And now we have this RNA that has to be cooked a little bit more before we can send it out to be made into protein. All right, I'm going to stop there because my time is up. We'll talk about RNA processing on Tuesday. Okay? So we covered a lot today, but it's the cool stuff. I mean, we're finally to the cool stuff. So hopefully it was not too fast. Ivan?
What is this still chapter 13?
No, we started 14. We finished 13 and we started 14.
Okay.
Marissa?
Are we going to go over the genetic disease project at all? Because from what I read on the rubric a little bit, I think I was able to look it over. I think it was a few days ago, and it seemed a little confusing to me. So I don't know if we're just on our own with that, or are we going to like...
So what it is you're going to pick a disease, and I want you to email me what you think the disease you want to study is. And basically, I want you to pick a disease that is genetically inherited from parent to child. Right? Because I want you to talk about that genetics, how that inheritance pattern, how does it pass on from one generation to the next. And then I want to make sure that it's something with a change in the DNA that ends up causing a change in a protein that changes cell function in some way that leads to the disease or the disorder, right? So, so pick a disease that you think sounds interesting, and then let me know, and then that's the way you're going to process the project.
Okay.
Yeah, but we had the discussion between a disease and a disorder earlier. I don't know that I really care if you come up with something that's called a disorder, as long as it has a heritable pattern so that it can be inherited from one generation to the other. Some disorders, for example, are not inherited, and so that's why I don't want you to necessarily pick that. But if it's called a disorder and it can be inherited, then you can just study it. If we want to, yes, you could research. Two of you could research the same disease for sure, but then you have to write your own paper. So you can research it together, discuss it with each other. That's totally cool. But then when you go home, you write your own paper. So this test covers 13, 14, 15, and 17. That's correct. But not all of 15 and not all of 17. So it's kind of half of those two chapters. And for those of you that came in late, the test is next week. I'm hoping Wednesday and Thursday. I might change that a little bit because I didn't get it as far as I wanted to today, so we might end up having a lecture on Thursday. And I might push the test back a couple days, but for right now, I'm planning on it for being Wednesday and Thursday. Discussion questions are already posted, and they're not due till Friday. Marissa.
So to check, because on the actual like assignment list, it says this presentation thing, and I know that's really not how this is going to happen. So, like, is it just basically skip the whole group part, and we're just writing this paper?
That's exactly right. We are just going to write the paper individually, and we're going to skip the whole group presentation part. So you'll just get your grade over the written part.
Okay.
Okay?
Okay. I think I'm good.
Okay. And if you have any questions, by all means, just email me. And what a good idea is just to tell me what disease you're thinking about. And if you want to like partner up with someone, email me the name of your disease. And then if someone else emails you that same, I'll put you guys together so you can talk about it. I'm happy to do that. When you say not all of the chapters, yeah, I think it's, well, let me pull up I don't remember exactly the names of the chapters. So hold on, I'll pull it up. All right, let's see. It is 13.1 to 13.3, 17.1 to 17.2, and 15.1 and 15.2. Can you see that right here? It's in an announcement, and it's also in the syllabus that's been updated. So if you look at the updated syllabus, it tells you what's on it. For those of you that came in late, if you turn in the vocab project late, that's okay. Cyrus, yes, I still want you to email me the disease that you choose, just because if it's one of those that doesn't have a good inheritance pattern, I want to be able to warn you ahead of time so that you don't get points taken off. No, there's going to be no presentation. Not for the genetic disease project. Which makes me super sad because it's like one of my favorite parts of the whole semester, but I just don't know how we would do it and cover all the material. It will be 100%, so it'll be worth whatever you get. However, I scored it on that sheet. I can't remember how I did it. Was it 30 points? Is that how I said I was going to do it? It will be whatever I give you. It'll just be a percentage out of 100. You won't have the presentation part. Make sure when you're doing it, you're paying attention right here. See where I'm pointing? Right here. Make sure you're doing those specific bullet points to fulfill your point getting. And the other thing is make sure you, when you're doing your your vocab project, that you look back at the rubric for that as well. Some of you have turned it in, but you didn't do all of the steps for every word. And you got to make sure you do, or you'll get points taken off.
The rubric is on the sheet for the vocabulary words, right?
It is. Yep. It's just on like the right-hand column of that sheet.
Okay.
It's in Blackboard. Any other questions? Some of you are going to be late for lab. Okay, I'll be around this weekend, so get your papers turned in. Don't forget. Work on your vocab projects, work on your genetic disease, study for the test. You can start your discussion questions, make it a biology weekend. And I'll see you on Monday for my Monday people and Tuesday for everybody. Okay? And I'll post the recording once it downloads. Okay, bye y'all.